So our next session is meningococcal vaccines, and to introduce this session, we have Dr. Stevens.
Good afternoon. I'm David Stevens. It's my pleasure to introduce the meningococcal vaccine session and also to chair the meningococcal work group. The subject this afternoon is MenB vaccines. One of the issues is revised dosing schedule for MenB-FHbp, Trumenba. Changes to the dosage and administration section for MenB-FHbp were approved by FDA on April 14, 2016. The original language was 3 doses according to a 0, 2, and 6-month schedule. The updated language is to administer a dose at 0, 1 to 2 months, and 6 months. Two-dose schedule administered at a dose of 0 and 6 months. And the choice in dosing schedule may depend on the risk of exposure and the patient's susceptibility to meningococcal serogroup B disease. So Heidi Suiters is going to talk about the impact of MenB-FHbp on meningococcal carriage. Dan Granoff from Children's Hospital in Oakland is going to talk about the immunogenicity studies of 2 MenB vaccines in adults, both as background presentations for the discussion. Laurel York is going to present on MenB-FHbp update, and then Jessica McNeil is going to present the considerations for use of 2 and 3-dose schedules of MenB-FHbp. There'll be an ACIP and VFC votes are anticipated. So the policy options for 2 or 3 doses, the 2 dose schedules of MenB-FHbp. For persons at increased risk for use during outbreaks, the preference for a 3-dose schedule at 0, 1 to 2, and then 6 months. When given to healthy adolescents, a preference for a 2-dose schedule at 0 and 6 months, or option for a 2 or 3-dose schedule, or the preference for a 3-dose schedule for healthy adolescents, as well as for those at increased risk. Of additional work group activities, a policy note regarding recommendations for the use of meningococcal conjugate vaccines in HIV-infected persons is to be published in MMWR on November 4th. The committee is continuing to review additional data on MenB vaccines and also considering the issue of booster doses for MenB vaccines for persons at increased risk. This is a list of the working group members. I want to thank those individuals, many of which are in the audience. And with that introduction, I'll turn it over to Heidi.
Good afternoon. Thank you for the welcome, and today I'll be discussing a meningococcal carriage evaluation that we conducted in response to a serogroup B meningococcal disease outbreak and resulting mass vaccination campaign that occurred at a college in Rhode Island. To give you some background and context, meningococcal disease most commonly takes the form of meningitis or bacteremia. It often starts with flu-like symptoms, but can progress within hours to a serious illness that can include high fever, severe headache, stiff neck, confusion, and a purpuric rash, as seen in this photo. This disease has a 10 to 15% case fatality ratio, even with appropriate antibiotic treatment. 11 to 19% of survivors have permanent sequelae, such as cognitive deficits or amputations due to necrosis of the extremities. Meningococcal disease is caused by the bacterium Neisseria meningitidis, which is a gram-negative diplococcus. Invasive meningococci generally have a polysaccharide capsule surrounding the cell, which confers the serogroup. 12 different serogroups of meningococci have been identified but only 6 primarily cause invasive disease. Traditionally, meningococcal vaccines are based on the capsular polysaccharide that is specific to each serogroup. Therefore, the resulting protection is serogroup specific. There are polysaccharide vaccines composed of purified capsular polysaccharide. Formulations include monovalent A, monovalent C, bivalent AC, and quadrivalent ACWY. There are also conjugate vaccines, where the capsular polysaccharides are conjugated to proteins such as tetanus or diphtheria toxoids. These vaccines achieve similar or greater immunogenicity and duration of protection than polysaccharide vaccines and are formulated as monovalent A, monovalent C, or quadrivalent ACWY. The conjugate quadrivalent ACWY, or MenACWY, vaccine is routinely recommended for adolescents in the US. This same approach of basing a vaccine on the capsular polysaccharides hasn't worked for serogroup B, because the serogroup B polysaccharide antigen is similar to human antigens, leading to poor immunogenicity and concerns about potential autoimmunity. As a result, serogroup B or MenB vaccines have to take a different approach and focus on outer membrane proteins. These antigens have multiple alleles and variable expression in different strains of the bacteria. Not all serogroup B bacteria will express the antigens that are present in a particular vaccine, and the serogroup B polysaccharide capsule, which is required for virulence, is not contained in the MenB vaccines. So unlike vaccines for other meningococcal serogroups, MenB vaccines are not expected to be protective against all serogroup B strains in circulation. Also, since they're based on outer membrane proteins and not on the serogroup-specific capsule, they may help protect against other serogroups as well. 2 MenB vaccines are licensed for use in the US, MenB-FHbp, or Trumenba, and MenB-4C, or Bexsero. MenB-FHbp is a 2-component vaccine containing an FHbp variant from each of the subfamilies A and B. It is given in 2 or 3 doses. Bexsero is a 4-component vaccine containing NAD-A, an FHbp from subfamily B, NHBA, and POR-A, and is given in 2 doses. In terms of transmission, meningococcal bacteria are carried in the human nasopharynx, and most carriers remain completely asymptomatic. Less than 1% of persons exposed to the organism develop invasive disease. The bacteria are spread through close contact, specifically via respiratory or oral secretions from patients or asymptomatic carriers. Important risk factors for both carriage and disease among adolescents include age, social mixing, and smoking. As the majority of transmission is thought to be attributable to asymptomatic carriers, decreasing carriage is the main way to provide herd immunity against meningococcal disease. Looking at what we know about vaccine impact on meningococcal carriage, in the UK, vaccines targeting serogroup C were shown to decrease nasopharyngeal carriage of serogroup C by 66% 1 year post-vaccination, thereby decreasing transmission and providing herd protection. MenA conjugate vaccine nearly eliminated serogroup A carriage. But our understanding of MenB vaccine impact on carriage is currently limited. The only previous study was conducted using MenB-4C and found an 18% reduction in carriage of any meningococcal bacteria and no effect specifically on serogroup B carriage by 3 months after completion of the 2-dose series. To date, no data has been published regarding the effect of MenB-FHbp on carriage. In the US, 3 serogroups, B, C, and Y, are the primary causes of meningococcal disease. This graph shows the incidence of these serogroups in adolescents and young adults following the introduction of the conjugate MenACWY vaccine in 2005. While incidence of all serogroups is low, serogroup B in black is the leading cause of meningococcal disease in this age group. In fact, serogroup B caused 7 outbreaks on college campuses since 2009, resulting in 41 cases and 3 deaths. To give you some background on this particular outbreak at Providence College in Rhode Island, 2 cases of serogroup B occurred in undergraduate students on January 31st and February 5th, 2015. The cases resided in different dormitories and had no known epidemiologic links. Both were determined to have a rare sequence type, 9069, never before seen in the US, and both case patients survived. These 2 cases, among a population of approximately 4,500 students, indicated an attack rate of 44 cases per 100,000, nearly 500 times higher than the national incidence among persons aged 17 to 22 years. In response to the outbreak, Providence College rapidly implemented a mass vaccination campaign using MenB-FHbp. The 3 doses were provided in February, April, and September of 2015, with remarkably high vaccination coverage of 94% with the first dose, 80% with the second, and 77% with the third. Incoming freshmen in the fall of 2015 were also offered to begin the series. CDC incorporated a meningococcal carriage evaluation into this vaccination campaign. It had 2 objectives: to determine the baseline prevalence of nasopharyngeal carriage of Neisseria meningitidis, and to assess the impact of MenB-FHbp vaccination on carriage. The evaluation consisted of a short questionnaire assessing risk factors for meningococcal carriage and disease, followed by an oropharyngeal swab. All undergraduate students at Providence College and graduate students who lived on campus were eligible to participate. Specimens were evaluated using culture, slide agglutination, real-time PCR, and whole genome sequencing. As I mentioned, 2 methods were used to determine the serogroup of each isolate: real-time PCR and slide agglutination. PCR is a genotypic test that detects the presence of capsular biosynthesis genes for each serogroup, regardless of whether or not they are expressed. PCR can detect serogroups A, B, C, W, X, and Y, and any specimen that doesn't contain the genes for one of these serogroup capsules is considered non-groupable. Slide agglutination, on the other hand, is a phenotypic test, which uses antiserum to look for expression of each serogroup, and can detect the same 6 serogroups as PCR in addition to serogroups E and Z, which rarely cause invasive disease. Any specimen that doesn't clump with the antiserum for any of these serogroups is considered non-groupable according to slide agglutination. Meningococcal bacteria can possess the gene for a serogroup but not be expressing them in their current environment, which would produce discordant results according to these 2 methods. Therefore, both methods were used in conjunction to get a more precise picture of meningococcal dynamics in this setting. Additionally, we abstracted meningococcal vaccination records for each participant, recording the doses and dates of previous Men-ACWY or MenB vaccination. This process was repeated 4 times, giving us 4 cross-sectional snapshots of carriage on campus. Round 1 occurred in February 2015, the week after the outbreak in Dose 1 clinics. Round 2 occurred in conjunction with the second dose in April. Round 3 occurred in September as upperclassmen received Dose 3 and incoming freshmen received Dose 1. And Round 4 occurred 1 year post-outbreak, including fully vaccinated upperclassmen as well as freshmen receiving Dose 3. For analysis, we performed descriptive statistics of participant characteristics. We looked at the proportion of students with overall meningococcal carriage, and specifically serogroup B carriage, and how this changed over time. We also estimated prevalence ratios using general estimating equation methods for repeated measures. Finally, as a number of students participated in multiple rounds, we're starting to examine within individual changes in carriage over time. The number of participants in each round ranged from 622 to 878. In total, 2,843 swabs were collected from 2,014 unique individuals. This table shows participant characteristics across the 4-round. As the evaluation took place across 2 academic school years, the class of 2019 was only included in rounds 3 and 4. The proportion of participants that lived on campus was high, but varied with each round. (34% to 42% of participants were male. Recent antibiotic use and recent upper respiratory symptoms were both highest during round 1, which took place during the winter. A high proportion of students smoked or had secondhand smoke exposure. And social mixing was common, as 67% to 74% of students reported visiting bars, clubs, or parties at least once per week. We also looked at meningococcal vaccine doses received at least 2 weeks prior to specimen collection to allow time to mount an immune response. A high proportion of students had documented Men-ACWY vaccine, generally received the summer before college entry. In round 1, no students had received MenB-FHbp more than 2 weeks prior. In round 2, most participants had received 1 dose. In round 3, most had received 2 doses. And in round 4, the majority had received either 2 or 3 doses. This table shows the proportion of students found to be carrying any meningococcal bacteria, and specifically serogroup B in each of the 4 rounds. In round 1, 24% of participants were carrying Neisseria meningitidis. This proportion remained fairly stable over the next year, with 24% carriage in round 2, 20% carriage in round 3, and 21% carriage in round 4. When looking at serogroup B carriage, 4% of participants carried serogroup B by PCR in each of the 4 rounds. By slide agglutination, just over 1% of students were carrying bacteria that expressed the serogroup B capsule in each round. This graph shows serogroup results by PCR over time. As with previous meningococcal carriage studies, most isolates were non-groupable, meaning the bacteria are unencapsulated and therefore better adapted for colonizing the nasopharynx. Serogroup B was the most common, and a handful of serogroups C, Y, W, and X were detected in each round. The next 2 slides show results according to slide agglutination. Again, the majority of isolates were non-groupable. Zooming in on the groupable isolates, we see that serogroup B was the most commonly expressed capsule, though serogroup E increased over time, and a handful of isolates expressed serogroup X, Y, C, and Z as well. Next, we looked at associations with carriage of any meningococcal bacteria. As compared with round 1, there was no difference for rounds 2 or 4, but round 3 had a slight association with decreased carriage. In comparison to the class of 2018, who were freshmen during the outbreak, the class of 2017 had the highest carriage prevalence. Male sex, smoking, and partying at least once per week were associated with increased carriage, while recent antibiotic use was associated with decreased carriage. Receipt of 1, 2, or 3 MenB-FHbp vaccine doses was not significantly associated with carriage. We then repeated the same models, but this time with the outcome of serogroup B carriage by PCR instead of any meningococcal carriage. We found very similar associations. During the evaluation, we found only 1 individual carrying the outbreak strain. They participated only in rounds 2 and 3 and were carrying this strain during both rounds. The carried strain was serogroup B by PCR, but was non-groupable by slide agglutination. Whole genome sequencing revealed that this was due to phase variation in the capsule locus, meaning the bacteria were temporarily not expressing the capsule. It's unclear whether this change in expression occurred in vivo or after the specimen was collected and cultured. In total, 615 students participated in multiple carriage evaluation rounds. 71% were not carrying any meningococcal bacteria during any round. 14% were consistently carrying meningococcal bacteria during each round in which they participated, though not necessarily the same strain or serogroup. 8% were carrying meningococcal bacteria during one round, but then were not carrying meningococci during a later round. And 7% did not have carriage, but then acquired carriage that was detected during a later round. Of the 50 students who lost meningococcal carriage, 13 lost carriage after 1 MenB-FHbp dose, 32 after 2 doses, and 5 after 3 doses. 45 students acquired meningococcal carriage, 20 after 20 after 1 dose, 16 after 2 doses, and 9 after 3 doses. Looking at serogroup B, 11 students lost serogroup B carriage, 2 after 1 dose, 8 after 2 doses, and 1 after 3 doses. And 10 students acquired serogroup B carriage, 3 after 1 dose, 4 after 2 doses, and 3 after 3 doses. In conclusion, in each round, 20 to 24% of students carried meningococcal bacteria, and 4% specifically carried serogroup B by PCR. This overall carriage prevalence is comparable to previously reported prevalences of up to 34% among university students in the UK. However, our observed carriage prevalence was quite a bit higher than recent US estimates of 1 to 8% among the general population. Despite this high carriage prevalence, only 1 carrier of the outbreak strain was identified, and no further serogroup B cases associated with the college have occurred. We did not find any evidence that MenB-FHbp vaccination impacts carriage at either the population or individual level. These results help inform US MenB vaccine guidelines, both for use in adolescents and young adults, and specifically in outbreak settings. Also, if MenB vaccines do not appear to affect carriage and therefore provide some herd immunity, this reinforces the need for high vaccination coverage during outbreaks to protect each individual and emphasizes the role of chemoprophylaxis for close contacts. Whole genome sequencing is underway to further characterize the isolates, compare carriage versus invasive isolates, examine within individual longitudinal data, and to investigate MenB vaccine antigens among the carriage isolates. Additionally, we conducted concurrent meningococcal carriage evaluations at 2 other universities, and we'll be comparing the findings from all 3 evaluations once they are complete. I'd like to thank all members of the Rhode Island Meningococcal Carriage Evaluation Team, which included staff from CDC, Rhode Island Department of Health, and Providence College. Thank you.
Thank you for that very nice presentation. Let's go on to the next, and then we'll have questions and comments. So if we could have Dr. Granoff.
Good afternoon. I appreciate the opportunity to be here to present some of our data on the immunogenicity of meningococcal B vaccines. So Heidi's already given a really good background. So we're used to thinking of meningococcal conjugate vaccines where there's a target, the capsule, and there's very little antigenic variation within a capsule group. So typically we measure bactericidal against one strain. And bactericidal activity is really the ability of antibody and complement to kill the bacteria, and it's an accepted correlate of protection. But with MenB vaccines and these MenB strains and the protein vaccines, we're dealing with a lot of antigenic variation, as Heidi already mentioned. So there's sequence variation and there's differences in expression. And as a result, these can cause resistance of some strains to serum bactericidal activity. So we're a bit limited on how broad the coverage would be by just measuring bactericidal activity to a limited number of strains. So for efficacy, the vaccines that were licensed were based on a limited number of reference strains, and therefore there's certain gaps in knowledge, which include the extent of protection against more diverse disease-causing strains, and then the effective vaccination schedule and duration of protection. So what I'm going to tell you about are results from 3 MenB immunogenicity studies in adults. And I have to start off saying they were not designed to provide comparison data between vaccines. Each one tested one vaccine. They were performed independent of industry. I have no relevant potential conflicts of interest other than I'm a inventor on patents related to meningococcal vaccines. I'm going to start with the conclusions just so that when you're looking at the data, you could be thinking about them. Essentially, 1 month post-dose 2, both vaccines elicited protective antibodies titers of 1 to 4 or greater against most strains. However, I'll show you some strains are relatively resistant, despite prediction that they would be susceptible by sequence analysis and antigen expression. And after dose 2, titers can decline within 4 to 6 months, especially for strains with low antigen expression. So study 1 was of MenB-4C or Bexsero. The 2 study sites, Oxford University and our group. It was a small study, 20 subjects. The median age was 29 years. Majority were healthcare or laboratory workers, and we gave 2 doses either at 0.1 or 0.2 months. Now you already heard that MenB-4C contains 4 components largely responsible for bactericidal antibody, factor H binding protein subfamily B, NHBA, NADA, and an outer membrane vesicle, which is predominant bactericidal responses directed at a specific antigen called P1.4. So a reference strain would be considered one that had absent, for example, NADA, a porin that did not have P1.4, and low expression of NHBA, and then you would be able to infer that most of the bactericidal activity was directed against factor H binding protein. So this slide summarizes data on 4-fold increases in bactericidal titer. So what we do is we take the titer 1 month post-dose 2 and divide it by the pre. So if a subject had, for example, a titer of 16 1 month after 2 doses and a titer of 4 before, then it would be 16 divided by 4, and that would be a fourfold increase. If it was less than that, it would be not qualified. And I'm showing data from the 2 studies that were done by the manufacturer for vaccine licensure that are in the FDA package insert, and our current study, which is shown in red. And for the 3 reference strains that we use for factor H binding protein, NAD-A, and POR-A, you can see that our data are very comparable to the data provided in the package insert. 95 to 100% are getting fourfold increases to the FHbp strain, the NAD-A strain, and about 70% to the POR-A strain, very similar to study 2 and higher than the manufacturer. So for at least our assays and our data for these reference strains are quite comparable. Now in real life, as I said, there's a lot of sequence variability. I'm showing you one way of looking at the sequences in subfamily B, factor H binding protein. If you have a single amino acid, you could be given a different ID number, so they're called peptide IDs. The MenB-4C vaccine contains ID1, and these are, in general, when you look at this, the closer this is, would be the similar in amino acid sequence and the further away, but they're all within the family and predicted to be covered by the vaccine. So we looked at bactericidal responses to a number of strains that were largely from outbreaks of disease on college campuses, UC Santa Barbara, the study you just heard from Rhode Island, Ohio University, Santa Clara, and Princeton, and then 2 strains from Quebec where there has been hyperendemic disease for a number of years. And I'm showing you the fourfold increases. These are all subfamily B, and these are the different ID numbers. And these other symbols, just to show the complexity, illustrate that these strains also had other antigens in the vaccine. So the asterisk refers to Neisseria heparin binding antigen, the little cross here refers to NAD-A. So this strain would have 3 antigens, factor H binding protein plus these 2 others. This strain has Neisseria heparin binding antigen, and I'm sorry I left this off the graph, but outer membrane protein, vesicle from the porin, P1.4. And so all of these had at least 2 antigens, and some of them had 3. And yet the fourfold increases were very similar to the reference strain for where there was an exact match with ID1 to the vaccine, but the fourfold are much lower against strains with other factor H binding protein variants. Now I want to really be clear that a fourfold response is a measure of a robust response. It doesn't say whether that person's protected or not. For example, if you went from less than 4 to 8, you would have a titer that's protective, but you would not be considered to have a fourfold increase. And I'm going to show you some data from Quebec, from the Quebec strain, where there was, 35% fourfold, and you'll see that most of the persons at 1 month post-dose 2 actually do have protective titers. So there's really 2 different measures, and my working model is that the proportion with 1 to 4 or greater is a good measure of short-term protection, whereas the fourfold response gives you a sense of the magnitude of the response, and it's likely to correlate with duration of protection. But again, this is a model. Now, these are the data for the same sera looked at a group of strains with factor H subfamily A. So here the factor H binding protein is mismatched, and we're depending on these other antigens of the vaccine, NHBA, this strain. This strain would have 2 antigens, which would be NAD-A and NHBA, and so would this one. And you can see it's pretty uniform, even for the strain that actually is mismatched for all 4 antigens. And you're getting around 30 to 40% fourfold increases in bactericidal titer. Okay, study 2 then is with MenB factor H binding protein in adults. So this is a totally separate study of the Trumenba. We have 2 sites, our own hospital and University of Massachusetts. The median age was a little older because we were dealing with all healthcare workers and laboratory people. And we used 3 doses at the recommended schedule at that time at 0, 2, and 6 months. And I remind you that there are 2 factor H binding protein antigens of this vaccine, subfamily A and subfamily B. The Pfizer nomenclature is B01, which is ID55, and A05 for the subfamily A, which is ID45. And you can see the relationship to a number of the other variants, and those are contained in the strains that we tested. So these are the data for 2 doses. I want to emphasize this is 2 doses of MenB factor H binding protein. Again, fourfold responses, 1 month post-dose 2 over the pre, the percent of subjects, and I made a little line at 70%. And you can see that for the subfamily A strains, many of them were more than 70%, whereas with the MenB-4C vaccine, most of it was at 40%. So it seems that having a subfamily A factor H binding protein may have an advantage for these strains. Now, we don't have the Pfizer strains in our lab to actually test, but I'm showing you the data in the package insert, and we actually find very similar type responses for the 2 doses of this vaccine to the 2 subfamily A strains that were used by Pfizer. Now, with MenB factor H binding protein, we give a 3rd dose at 6 months. So these are the data showing the 4-fold responses after 2 doses and 3 doses. The 3 doses is the 1 month post-dose 3 compared to the pre. And with the 3rd dose, you can see that there's a significant increase for many of the subjects and for many of the strains, and most of them are in the 80 to 90% 4-fold increases. And again, our data are similar to what we reported for the subfamily A. Now for subfamily B, I'm showing you now data for 2 doses at the 0-2 schedule. And you can see quite a lot of variability. I'll just make one point here. This strain, happens to be a mutant that we included, and this is a low expressing mutant, about 50% lower expression than in the wild type. And you can see as you have lower expression, it really does affect the ability of the antibodies elicited by the vaccine to give you a robust bactericidal response. And these are the various university strains, and 2 doses are giving within the same range as the MenB-4C. And I have the Pfizer data here, which are very similar to what was reported for the subfamily B strains. And a 3rd dose given at 6 months, you get a significant increase for many of the strains, almost all of them over 70% 4-fold. And again, the data are quite similar to what was reported. So I'm only going to show you one strain in detail. For the committee, there's some backup strains if you want to look at the data in your handout, where we're actually going to show you now the titers of persons who are likely to benefit most from vaccination, the ones who have titers less than 1 to 4 before vaccination. So this is data against the Quebec strain from MenB-4C. So we had 13 subjects in the group of 20 that had titers less than 4. And you can see that although most of them are not getting a 4-fold response, 10 out of the 12 that we had, it should be 11 out of 13, had protective titers after dose 2. But we have a small number of subjects who had samples obtained 4 months after post-dose 2. So you can see they tended to decline. So when you have low titers, it seems like they decline to below 1 to 4. For MenB factor H binding protein, you have very similar results. After 2 doses, 13 out of 16 had titers of 1 to 4 or greater. And then there was a decline in titers. But with that vaccine, you give a 3rd dose and you get a booster, and 11 out of 12 after the 3rd dose have protective titers. And we don't have follow-up sera on these at this point in time. Okay, so I'm going to switch now to a different kind of study, which is the last one, and it's called Study 3 of the Meningococcal 4C immunogenicity in Santa Clara University students. So in January 31st to February 3rd, there were 3 cases, 2 culture confirmed and 1 suspect, caused by a strain that it was in the ST32 complex. And within a few days, they organized clinics at the university, and 4,921 persons received a 1st dose of vaccine over a few days. And then in March 18th and April 6th to 8th, they had additional clinics, and 4,731 persons were immunized. Most of them got the 2nd dose. A few of them that had not gotten the 1st dose got the 1st dose at this point in time. We collected sera over a few days in May from 246 students. There were 52 who had no vaccination, 91 who had gotten 1 dose, and 101 who had gotten 2 doses. So the main comparative group here really is 0 doses and 2 doses, in which we're getting samples somewhere between 6 and about 11 weeks after post-dose 2. And we measured bactericidal against 3 college outbreak strains, Santa Clara, Princeton, and Ohio universities. So these are the data for the prevalence of serum bactericidal activities at this time point. I want to emphasize it's an interim analysis. We're actually totally blinded, so we are measuring the serum not knowing the number of doses that we got. But we sent the results to the statistician, Dr. Liu, at Worcester Institute, and she provided this analysis for this meeting. Again, the percent now are with subjects who have titers of 1 to 4 or greater, which would be considered protective, and a higher response of 1 to 16 or greater. And keep in mind, for a 4-fold, you need a minimum titer of 1 to 16. So it gives you some idea that you would be about a 70% 4-fold response in the 2-dose group. Interestingly, against this strain, around 40% already had protective titers before vaccination, and 20% had high titers. After 1 dose, it increased, and after 2 doses, we had 92% had protective titers. That would be about 1 to 2 months after the second dose, and about 60 to 70% had high titers greater than 16. Now, we also measured, as I said, to the strain causing the outbreak at Princeton University, where there were 9 cases and 1 death. This strain expressed 2 MenB-4C antigens, factor H binding protein subfamily B and NHBA. And a previous study that you, I'm sure, are aware of in the New England Journal by Boston and colleagues showed that 66% of the subjects, students immunized with 2 doses, had developed protective titers of greater than or equal to 1 to 4. So these are the data from looking at the Santa Clara University sera against the same, it's actually the blood isolate of the same patient that they studied with using the CSF isolate. And first you see that the 0 doses, the prevalence of background antibody is much lower. Almost nobody had a titer of greater than equal to 1 to 16. There's a significant increase after 1 dose. And after 2 doses, we found 69% with titers of 1 to 4 or greater. So we virtually replicated the data that they had done at Princeton University, indicating that this strain is relatively resistant, especially when you compare it to the 92% protective titers against the Santa Clara University strain. Now, finally, we looked at a outbreak strain from Ohio University, where there had been 13 cases and 1 death. The outbreak strain expressed 3 MenB-4C antigens, factor H binding protein ID15, NADA, and NHBA. And these are the data, again, from the interim results. You can see that the background level of antibody is very low in the Santa Clara students against this strain. And after 2 doses, around 52% had titers of 1 to 4 or greater, and around 10% had titers of greater than 1 to 16, despite the fact that there was a match in the antigen and the expression of MenB-4C, factor H binding protein, NADA, and NHBA. Okay, so to conclude about these vaccines, first, they are complex, at least 4 antigens, and as I mentioned, there's probably other antigens in the OMB that could contribute. The antigen-specific reference strains that were used as a basis for licensure are more susceptible to the vaccine-induced antibodies than many clinical isolates causing these outbreaks. Some strains are relatively resistant to bactericidal despite prediction of susceptibility by sequence analysis and antigen expression, and the Princeton strain and the Ohio University strain would be one. I want to emphasize, I haven't shown you the data, they're in your handout, that the great majority of subjects to most of the strains had titers of 1 to 4 at 1 month post-dose 2. But again, some of the resistant strains, those antibodies declined, especially when they were low-expressing strains of some of the antigens. So the MenB factor H binding protein, the 2-injection data I think are the most interesting, because we basically saw very similar, in some cases, like to the subfamily A, the factor H subfamily A strains even a bit higher, fourfold increases in bactericidal as in the MenB-4C. The great majority of subjects, again, though, even the ones that had resistant, were more than 1 to 4 at 1 month post-dose 2. Titers declined again against the low factor VIII binding expressing strains, and the recommended third dose at 6 months boosted titers. We clearly need additional data on antibody persistence, which should include, I think, some relatively resistant strains to get sort of a sense of just what the variability is in antibody persistence. I'd like to end really on the limitations because I think that's important. I want to emphasize that these were independent studies that were not designed to compare two vaccines. The populations are different. One used UK students largely for the MenB-4C, although we do have a lot of now US data on the Santa Clara University students. We have relatively few sera 4 to 6 months post-dose 2. That was really added at the end of the protocol, and we couldn't get everybody. The serum bactericidal assays use research assays, and they were not the type that would be FDA validated, although the assays are robust. And the data on the Santa Clara University study are really on antibody prevalence. I don't have pre-vaccination sera, but we can compare the 0 dose to the 1 dose and the 2 doses. And I want to emphasize this is an interim analysis that requires much more work. And then a lot of people, and I'm not going to go through, worked on these studies. I really want to thank CDC, that the Meningococcal laboratory providing these strains so that we could use them as test strains. Sanjay Ram at the University of Massachusetts, who collaborated in the MenB-FHbp binding protein assay, and Andrew Pollard's group in the UK at Oxford. And, yep, can't go backwards. Yeah. And finally, the Santa Clara University involved quite a few people, particularly Kathleen Winter, who was really pivotal in organizing the study with the group in the health department and the university, and Eduardo Luzon, who performed almost all the bactericidal assays in this study and the others. I'll stop. Thank you.
Thank you very much. Do we have questions for Dr. Souters or Dr. Granoff?
Well, my name is Rino Rapoli. I'm from GSK, and actually I'm the one who's been kind of inventor of Bexero. First of all, thank you, Dan, for the presentation. I think it's very well done, and I only wanted to make a few comments. And I think, as you highlighted, the two vaccines, Bexero and Trumenba, are quite different. They are different antigen compositions. Basically in the way we deliver the number of doses. And I think it's important to keep that in mind because Bexero has 4 different antigens targeting completely different antigens, and each of them can kill the same bacterium. So I think it has multiple hits on the same thing. And one thing I noticed in your presentation, which is actually pretty interesting, is that in the Trumenba, you have, when you go between the 2 and 3 doses, you have a pretty good increase in immunogenicity and coverage. That is more or less what we observed with Bexero between 1 and 2 doses. And in Bexero, we licensed Bexero as a 2-dose after multiple studies. And in one case, during the phase 3, we also did 3 doses, and we did not see that kind of increase. So in Bexero, the increase in number of protected people increases between 1 and 2, while it looks like for Trumenba increases between 2 and 3 doses. The other thing I think you mentioned, but it's important to remind, that you showed the 4-fold increase, which I tend to agree with you is a way to look at maybe the longevity of protection, but it's also very important to remember that that may underestimate the number of really protected people, because many people may have a protective titer above 1 to 4 in bactericidal assay and still not have a 4-fold thing. So I think the comment that you made that this basically, the people who do not have a fourfold increase may still be protected, I think is a very important one. We need to look at that number. Then the other thing I wanted to mention is that obviously all these serological things are very important, but they are always a surrogate for real data in the field. And that's what we've been missing during the development of vaccines for so long. In the case of Bexsero, I think we have two types of data. One coming from multiple usage and outbreaks in United States and Canada, where 2 doses basically have been used in many instances. They're not being breakthrough cases. That's very, very important to remind. So we have not seen cases in all the vaccinated people. And the other one, which is the piece of data that came from the United Kingdom. Bexsero has been used in infants in United Kingdom to vaccinate all the newborns starting September 2015, more or less a year and a month ago. 2 doses were used in infants. Now, as you know, infants are completely naive to meningococcal, so they are both difficult to protect in adolescents. In spite of that, 2 doses were used, and with 2 doses, we got an efficacy which was reported by Public Health England a month ago in a meeting in Manchester. They report an effectiveness of 83%. So basically, with Bexsero, we got effectiveness in the field in a very difficult population, which is extremely high. So I just wanted to share these things with you and with the panel. Thank you.
Thank you. Other questions? Yes, Dr. Walter.
For our first speaker, I noticed that the coverage for vaccination declined from 94% for the first dose to around 80 to 77 for the second and third in the campaign. Was some of that, do you have any sense of why that was? Was it just at the start of the campaign people were concerned about disease, or was it due to side effects and concern about side effects?
I think, well, I'll just say overall we've seen a drop-off in these MenB vaccine campaigns at a lot of the colleges that have had outbreaks. In this particular case, the school did something unusual, which is that they required all students to come to the vaccination clinics and they could sign declination forms if they didn't want one. So I think that's how they achieved really high coverage during the first round. They did continue to require it, but I think students kind of caught on that if they didn't show up, something didn't happen to them. So they had some sort of drop off over time. We didn't hear many, I mean, all the students, a lot of them experienced and mentioned sore arms, but I didn't hear students saying that that was a reason that they did not come back to get vaccinated. But at some of the other schools, we're doing some more surveys asking questions about students' opinions about the vaccine and what's influencing their decision to get vaccinated or not.
Thank you. Dr. Friedland.
Yes, Dr. Friedland from GSK. Also about the first presentation, I'd like to congratulate the Rhode Island Meningococcal Carriage Group for undertaking such an important study. Certainly, the evaluation of the impact of meningococcal B vaccines, Trumenba and Bexsero on carriage is very important. At GSK, we're committed to continue to evaluate the impact of Bexsero on carriage. We're in final stages right now of finalizing a protocol to evaluate the impact of Bexsero on carriage in over 30,000 individuals that will be powered to detect even small differences in carriage, which we think is very important. And we look forward to sharing these data with the committee as they become available.
Thank you. Who else? Dr. Belongia.
Thank you. I wonder if you could just clarify a little bit about the titer of 1 to 4 as a protective titer. Can you tell us a little bit more about the basis for using that as a correlative protection?
Yeah, that's a really, it's a good question and a long history for those who are not in the meningococcal field. But it goes back to Goldschneider study in the 60s, I think, and then published in the Journal of Experimental Medicine, in which they looked at military recruits. So they got serum samples at the time they started training, and it was a serogroup C outbreak. And then there was quite a few cases that occurred over the next few months. And then they retrospectively took the sera from the people who got disease, and then a sample of around 500 sera from people who didn't get disease, and a titer of 1 to 4 or greater, because they only took a single dilution of 1 to 4, essentially gave you about 95% protection against developing serogroup C disease. And then if you look at the people who lacked titers, so essentially almost all the cases were in people who lacked titers. They also had a colonization study going on, and they detected, I don't know, maybe 30 carriers. It's a long time since I read the paper, but about a third of those actually went on to get disease. So there's a really, at least with serogroup C, and this was natural antibody, it wasn't vaccinated. If you have a titer of 1 to 4, it really correlated with protection. And then there's many other studies that have inferred 1 to 4, looking at what's happened after introducing vaccines and trying to make the link for serogroup B and for these vaccines. Suffice it to say that collectively the data are pretty convincing that a titer of 1 to 4 does predict protection. A titer less than 4 doesn't necessarily mean you're susceptible, but was associated with all those cases. And this has been accepted then by regulatory authorities all over the world, including the FDA, as the basis of licensure. These vaccines were not tested in efficacy studies. They used the 1 to 4 and the 4-fold as a composite to look at the immune responses, and that was the basis of deciding vaccination schedules and inferring efficacy for the vaccine.
Thank you. Dr. Paradiso?
Peter Paradiso, I had a question about the carriage study. It seems that often we when we look at a bacterial carriage in the nasopharynx, it seems like people generally consider that clearing the bacteria is totally different than affecting acquisition of new bacteria. And often when we see these carriage studies, it's really looking at blocking the acquisition of new bacteria. And I saw that you did separate those out in your study, but the numbers were very small. But on the other hand, in these kids who are 18 to 22 years of age, 20% of them or more had carriage that they had gained somewhere along the way, and probably based on the epidemiology when they were 14, 15, 16 years old. And so I guess I'm wondering whether there's an opportunity or whether there's any plan to maybe back up in the age group and look and see if you can stop acquisition in a younger age group before it builds up to the 15 or 16-year-olds, or if that's even feasible to do.
We don't have any current plans to look at carriage in adolescents. I will say that we're starting to dig into the within individual dynamics in carriage over time in this particular population, and it seems like some of the students can carry for a couple of weeks, some can carry for a couple of months. We're trying to see if they're carrying the same strain or if it changes. So I'm not sure if they're actually acquiring some of these strains in adolescence and then bringing them to college with them, or if there's just a lot of circulation and transient carriage happening. We did look specifically at the freshmen, since the freshmen the year after the outbreak, because they came into campus the year after the outbreak, and they did have an increase of carriage. We saw them their first week on campus, and then again 6 months later, and we did see an increase in carriage in those incoming freshmen.
Yeah, and I thought it was also interesting that these two vaccines are not really group specific completely, because they're protein antigens, and looking at all of those strains for which you couldn't assign a group, you actually saw a little impact on that group, and that may be pointing towards a trend. I don't know that, but just a comment.
Thank you. Dr. Maldonado, and then I think we'll move on.
Thank you for those really elegant presentations. I have a question about the historical colonization that we understand from the 50s, looking at some of the Army studies done in California and other places, and looking at baseline colonization and what happened. Those are very, very nicely done natural history studies there, and also the impact of antibiotic prophylaxis and resistance. So can you compare some of, I'm not asking you to do this right now, but is there a possibility of comparing the data from those studies to what you see now? Obviously you have better genetic data now, but there seems to be a lot of parallels here around what happens to these people, and more importantly, we were just talking about, if you knew somebody was carrying, what would you do with that patient? Because what was shown in the Army studies is that it didn't have a huge impact, but you wound up with a lot of rifampin resistance.
That's not something that we've looked at, but we definitely can look and compare to some of those earlier studies from a long time ago.
Maybe I could just say that actually
Yeah.
there's a variety of literature on this, including studies that David Stevens and I tried to do in adolescents and high school students 10 years ago, getting really low carriage studies. And so I think this is a really complicated field, and we're very much looking forward to the data the pharmaceutical company is doing with much, much higher enrollment rates. I think the studies that CDC have done, when we tried to do it prospectively, we've had sample size power problems. When we tried to do it in convenience around outbreaks, there are all the problems that are brought up here. And so I think this really large study that's being planned may help. David, do you want to?
Thank you. So let's move on. Dr. York.
So while we're waiting for Dr. York, if I may, just to comment, I think one of the issues is that we now have a lot better typing systems and understand very specifically, for example, in the study, only one individual was carrying the outbreak strain, and I think that's quite a difference than what we knew about in those old military recruit studies.
Right. Thank you very much. I want to thank the work group and the ACIP for the opportunity to provide an update on Trumenba. So as was already stated, Trumenba was licensed in October of 2014 for use as a 3-dose series, and you'll recall that this was really an accelerated approval in response to the unmet need with these outbreaks that we were seeing occurring in the U.S. We continue to discuss the schedules with FDA, and on April 14th, the FDA approved a label change under those same accelerated approval regulations. And we now have a flexible 3-dose schedule at 0, 1 to 2 months, and 6 months, and a 2-dose schedule at 0 and 6 months. And both schedules were determined to be safe and effective by FDA. And so what I'd like to, has been said to you, is that with this new scheduling that we have, or to the 2 different schedules we have in the label, that we really do need to have ACIP guidance in reference to the existing recommendations, and that's needed for providers as they move forward with use of these vaccines. I'm going to briefly go through the data that supported the change in the label, but I'll also provide to you some new data on persistence and immune responses following a booster, and be able to look at those in terms of both schedules, the 2-dose schedule and the 3-dose schedule. You'll recall that Trumenba is a vaccine that's, as we all know now this afternoon, based on a surface-exposed factor H binding protein, or FHbp. This protein functions as an important immune evasion mechanism for meningococci. In binding factor H, it actually downregulates the complement cascade and limits the bactericidal activity of complement and that membrane attack system. This protein is expressed on greater than 97% of the invasive MenB strains that we've looked at, and there's been a comprehensive evaluation survey of meninge isolates from 2000 and on, in fact, and these are invasive disease isolates in the US as well as several countries in Europe and in Canada. The FHbp sequence is segregated into 2 genetically and immunologically distinct subfamilies that we refer to as subfamily A and B, and Dr. Granoff, thank you for presenting that portion for us. And Trumenba is in fact composed of 2 lipidated proteins, and the lipidation of that protein is important and really contributes, at least being able to look at this in preclinical studies, really contributed to that breadth of protection that we saw, the ability to elicit antibodies that could recognize across the diversity of this particular antigen, and recognizing what I mean in terms of recognizing across the subfamily and the variants within that subfamily. The vaccine-elicited protective responses are actually demonstrated not through efficacy studies because of the disease incidence that we have with meninge, but through the correlate that we use to predict protection, and that is the serum bactericidal assay using human complement, We use 4 MenB strains that are representative of prevalent strains in the US and in Europe, and these are invasive disease strains. They are representative. They express FHB variants that are different from what we have in our vaccines, so really ascertain the ability of antibody to recognize across diversity of those components. And this approach is really designed to demonstrate the breadth of coverage. Where those 4 strains are representative, we would expect them to predict what you will see against the activity against other strains. The study that contributed to being able to have 2 schedules and this change in the dosing administration is from a study that was conducted in Europe. It was individuals from 11 years through 18 years were randomized into 5 groups so that we could look at 2 3-dose schedules and 3 2-dose schedules. The participants were in fact enrolled into an additional study or a follow-on study with a different number, and this was to evaluate the antibody persistence. This was evaluated annually, and then we looked at the response to a booster dose at approximately 4 years. The choice of that timing was really not in terms to be able to determine when to give a booster dose, but really to look at the ability to boost the response, to look for memory. And we knew that these data would be important to ACIP and their considerations of use of the vaccine, and so the timing for us to have these data were really based on being able to provide additional information for your considerations. This table really will show you the benefit of Trumenba and also what happens with interval, the effect of interval on providing vaccine. As I said, the immune response will be evaluated on the 4 representative strains, and we look at 4-fold responses, the percentage of individuals who have the 4-fold response. And as Dr. Granoff said, this is really looking at the vaccine response and the robustness of the response. The minimum titer that a person has to achieve is 1 to 16 to be recognized as a responder with a 4-fold response. We also look at a composite response, which is looking at the ability of an individual, the antibody generated in that individual, to actually recognize each one of those 4 strains all together, so a combination. And so this really does show the ability of the antibody to recognize across the diversity of the antigens. You'll also remember that I've already shown you data in terms of additional supplemental data on 10 additional strains. When I talk about the prediction of what we see on those 4 strains, it was then we provided additional information, supportive supplemental information on additional strains to really highlight how these data do demonstrate the immune response that's elicited by Trumenba. If we look at the immune response in terms of 2-dose schedules, and if we go from the right over to the left, and you can see that we have a number of different intervals, a 0-1, a 0-2, 0-4, and 0-6. The 0-1 is really coming from that 3-dose schedule, looking post-dose 2 on a 0-1-6, but this is really to be able to show you the real effect of changing the interval in providing 2 doses. And you can see that in terms of 4-fold, but also the composite response, that as you increase that interval, you're getting an improvement on the immune response that you actually acquire from receiving the vaccine. And this was really important in terms of the 0-6 interval providing robust responses and substantial protective responses in terms of the 4-fold response, but also in terms of the composite response. And you can see here in terms of a point estimate of 73.5. So a nice, robust, substantial protective immune response with a 2-dose interval that's 6 months. There's also, if we look at 3-dose schedules, there are circumstances when we want to give 2 doses in close interval. So in those circumstances, for example, like an outbreak, we want to get an immune response as quickly as we can and in a high number of individuals as we can. And so we know from the 3-dose schedule that we can use a 0-1 or 0-2 and get a comparable type of response, but in fact there is an improvement in terms of the percent of responders that we see with that 3rd dose in the 3-dose schedules. And so I think that Dr. Granoff's data shows this very clearly in terms of a 2-dose schedule, which is at a 0-2 or a 0-1. There is an immune response. It's a very effective immune response, but if you want to have a high proportion of individuals that are going to respond and have a broadly protective response, that 3rd dose will provide it in the 3-dose series. We also see this trend if we look at the response in terms of the percent of subjects who have an SBA titer of greater than 1 to 8, and in fact 1 to 16 on one of our test strains. Again, this is higher than the protective correlate of 1 to 4 that we accept from those studies that were done very early in the military. And again, you can see the effect of that interval, the range of response that we would expect to see providing an interval, a very short interval versus a longer interval of a 2-dose series. And then the 3-dose series also giving you a robust response, a high percentage of individuals, and a high range of response that we would predict to be seen against other strains, diverse strains. This is also seen in terms of the GMT with the 6-month interval giving you slightly higher responses in terms of GMT, more comparable to in fact, the 3-dose series. And you can see that for some of these responses in terms of the percent of individuals with a titer greater than 1 to 8 or on the GMT that there are overlapping confidence intervals. And we expect these 2 schedules now, as I said, in terms of effectiveness, and this is why that we have these 2 schedules now in our label. In terms of the safety, I had covered this with you before. There's really no differentiation between the 2nd or the 3rd dose, and so consequently, again, as I said, FDA approval in terms of safety and efficacy, effectiveness of these vaccines, of this vaccine on a 2-dose schedule 0-6 or a 3-dose schedule of 0-1 to 2 or 6 months. So now I'd like to present to you the persistence data that we have following either a 0-6 or a 0-2-6 schedule of Trumenba. And this is being shown to you in terms of the percent of subjects with an HSBA titer of greater than 1 to 4. There are 2 cohorts being plotted for you against the 4 representative strains. So you can see the 0-6 is in the blue line and the 0-2-6 cohort is shown to you in the gray. That there is, as we've seen before in a previous study that's been discussed with the work group and presented to ACIP, that there is a plateau that's established after a waning of the persistence in terms of the percent responders. And there seems to be a consistent plateau over the 48 months for Trumenba. There is a divergence with 2 of the strains, as you can see here, but the confidence intervals do overlap. And this would say to us that the response that you have after a 2-dose schedule or a 3-dose schedule will provide persistence in terms of the protective response. There is a lowering of the response in terms of the percent or the range that you will see against diverse strains, but clearly there's population, in terms of the population, there is still a protective response against MenB. It was interesting for us to look at a booster response, the HSBA responses following a booster about 4 years out, either having individuals having the 2-dose schedule at 0-6 or a 3-dose primary schedule at 0-2-6. And for simplicity, I'm showing you here the response in terms of the the post-primary series, and then 48 months post that primary series, so the data that you've seen without all those additional time points from 12 months through to 48. But to make a point in terms of the booster response that's seen against these 4 representative strains, in fact, showing a significant percentage of individuals who are responding to a single booster dose. And if we look at the composite on this, we are now having a composite that's well over 90%. So clearly there's been induction of memory, and probably approximately the same level of memory on either the primary series of a 0-6 or a primary series of 0-2-6. The memory response is also well defined and shown in terms of the HSBA GMT responses to the booster dose, and you can see the titers in terms of the the GMT at 0.6 in the blue. Again, the GMT is 0.26 in gray. And that we have significant responses is in terms of GMT after primary series of the 2-dose or the 3-dose schedule. Maintaining, as I said, that plateau in terms of the persistence above the protective level of 1 to 4. And then post-booster, we're having a significant increase in terms of the GMT, which really demonstrates that memory response. In terms of the safety of a booster dose, we don't see a difference in terms of the safety profile, so there's nothing that's untoward in terms of receiving a booster for approximately 4 years post your primary series. So in summary, the FDA approval under accelerated approval regulations, we now have a flexible dosing schedule for Trumenba, a 3-dose schedule at 0, 1 to 2 or 6 months, and a 2-dose schedule at 0, 6, and FDA seeing these in terms of being safe and effective. So either vaccine can be utilized to provide protection against invasive group B meningococcal disease in individuals 10 through 25 years of age. These data demonstrate that both schedules provide a significant benefit in terms of the breadth of coverage against diverse MenB strains causing disease. And there's the flexibility within the 3-dose may be beneficial when the individual is at significant risk. For example, in an outbreak, we want to give 2 doses in a very short interval to maximize the response quickly. And then the 2-dose schedule may improve the compliance in an age group where the completion of a multi-dose series is challenging. From the data, we can say that there's a similar persistence in the HSBA response, and clearly after either series, the immunological memory is established in a booster dose elicits a response that's actually higher than the primary response after the primary series. Thank you very much, and I'd be very happy to take questions.
Questions for Dr. York? Dr. Lee?
Just one quick question. When you received the FDA labeling and both schedules were determined to be safe and effective, at what time point was the effectiveness assessed? Was that 1 month post?
This
the
is
last
always
dose?
1 month after the primary series, yes. And all the vaccines are licensed on that.
And are there long-term follow-up studies that go beyond what you've shown us here?
Not planned. We will, in fact, have another bleed in terms of after the booster dose, so we'll be able to come back with you with some data about a year out from that booster dose. So we'll see if there's the waning is less and then plateauing within that.
Other questions? Seeing none, Ms. McNeil, on to considerations.
Thank you.
I will now provide a summary of the work group's considerations for the use of 2- and 3-dose schedules of MenB-FHbp or Trumenba. As a reminder, there are 2 serogroup B meningococcal vaccines licensed for use in the United States in persons 10 through 25 years of age. MenB-FHbp contains 2 components and was originally licensed by FDA in October of 2014 for administration as a 3-dose series. MenB-4C contains 4 components and was licensed by FDA in January 2015 for administration as a 2-dose series. MenB-4C is also licensed in a number of other countries for use in persons 2 months of age and older. The current ACIP recommendations for the use of serogroup B meningococcal vaccines are that people 10 years of age and older who are at increased risk for meningococcal disease should receive MenB vaccine, and that adolescents and young adults aged 16 through 23 years may be vaccinated with a MenB vaccine to provide short-term protection against most strains of serogroup B meningococcal disease. In both of the MenB policy notes, additional guidance is provided that states that the MenB vaccine should be administered either as a 3-dose series of MenB-FHbp or a 2-dose series of MenB-4C, consistent with the original licensure of these vaccines. On April 14th, the FDA approved updates to the dosage and administration section for MenB-FHbp. Those changes are highlighted in blue below, and they allow for administration of both a 3-dose schedule at 0, 1 to 2, and 6 months, and a 2-dose schedule at 0 and 6 months. Additionally, the package insert now includes the following statement: the choice and dosing schedule may depend on the risk of exposure and the patient's susceptibility to meningococcal serogroup B disease. Today, I will summarize a work group's interpretation of the short-term immunogenicity data, antibody persistence, and safety data for the 2 and 3-dose schedules of MenB-FHbp, and then I will summarize the proposed policy option language and the work group's discussions. As a reminder, several dosing schedules were evaluated for MenB-FHbp, including 2 3-dose schedules and 4 2-dose schedules. Today, I will focus on the data presented for the 2-dose schedule at 0 and 6 months, along with the 2 3-dose schedules. Okay. Based on the short-term immunogenicity data, or immunogenicity 1 month following the last dose, among the 2-dose schedules evaluated, the 0-6-month schedule had both the highest proportion of responders in GMTs and is most similar to a 3-dose schedule. However, the proportion of subjects demonstrating a composite response to all 4 primary strains is slightly lower with the 2-dose schedule at 0 and 6 months compared to either 3-dose schedule. Similarly, for most strains, the GMTs are lower with the 2-dose schedule at 0 and 6 months compared to either 3-dose schedule. For some strains, the 95% confidence intervals around the GMTs do not overlap. Antibody persistence to the 4 primary strains is shown here for participants who received 2 doses at 0 and 6 months or 3 doses at 0, 2, and 6 months. The percent of subjects with HSPA titers greater than or equal to 1 to 4 to the 4 primary strains is similar through 48 months for adolescents who received either 2 or 3 doses of MenB-FHbp. In addition, the GMT responses to a single booster dose 4 years following receipt of either 2 or 3 doses of MenB-FHbp are similar. Finally, as has been presented to ACIP previously, the MenB vaccines are generally more reactogenic than other vaccines given during adolescence, and the most common adverse event reported is pain at the injection site. But overall, the safety and tolerability profiles are similar for both the 2 and 3 dose schedules of MenB-FHbp. So again, there are 2 serogroup B meningococcal vaccines licensed in the United States for persons 10 through 25 years of age, and ACIP does not express a preference for either vaccine product. The current ACIP recommendations for the use of serogroup B meningococcal vaccines are shown here, and no changes to those recommendations are being proposed today. However, because of the recent change in licensure for MenB-FHbp, during the remainder of our discussion today, we will focus specifically on MenB-FHbp in order to provide guidance from ACIP on when the 2 and 3 dose schedules of MenB-FHbp should be used. After reviewing the data, the work group discussed several different policy options. First, for persons at increased risk, including during outbreaks, a preference for the 3-dose schedule of MenB-FHbp is proposed. For healthy adolescents, there are 3 possible policy options: a preference for a 2-dose schedule, an option for either the 2 or 3-dose schedule, or a preference for the 3-dose schedule. Based on the discussions of the work group, there was agreement that ACIP guidance for which schedule to use in each population is needed. There was strong consensus in the work group to express a preference for the 3-dose schedule for persons at increased risk, including during outbreaks, to provide early protection and to maximize the immune response. For healthy adolescents, the work group's considerations included the following. When considering either a preference for the 2-dose schedule or an option of either the 2 or 3-dose schedule, the key considerations were that the persistence of the HSPA responses following the 2-dose schedule was similar to the 3-dose schedule, and the GMT responses following a booster dose were also similar. However, the work group did note that providing an option for either the 2 or 3-dose schedule does not provide explicit guidance to the physician on which schedule to use in healthy adolescents. When considering a preference for the 3-dose schedule in healthy adolescents, the key considerations were that for people who want to maximize short-term protection, 3 doses is preferred. Both the 2 and 3-dose schedules take 6 months to complete, And ACIP could provide guidance that if someone receives a second dose of MenB-FHbp 6 months or more after the first dose, no additional doses would be needed. Based on these considerations, the strong consensus of the work group was to express a preference for the 3-dose schedule for persons at increased risk, including during outbreaks. In addition, the strong consensus of the work group was to also express a preference for the 2-dose schedule for healthy adolescents. And I would now like to open this session up to questions and discussion. Thank you.
Thank you. Questions for Dr. Walter?
Yes, in the work group deliberations, was there any discussion about harmonizing the schedule to 0 and 6 months for both vaccines when given to healthy adolescents?
We did not discuss that, but just was basing this on FDA's licensure of the 2 vaccines. But that's something we could consider. Yes.
Dr. Atmore?
The data you showed us showed clear differences at 7 months, but in terms of the early protection, did you all have data that you didn't show us about immunogenicity or serum antibody levels at earlier time points? In other words, other than the benefit at 7 months, was there a benefit to receiving an extra dose at 1 to 2 months, say at 2 to 3 months, compared to not getting that dose?
I mean, I think the data shows that the longer the interval between the doses, the better the response. And so, I mean, I think after 1 dose, you get a better response after a longer interval than a shorter one.
Yeah, but that wasn't my question. Okay. My question is the early protection, to me, implies within several months, as opposed to at the 7-month time point, which is where we were shown that there were clear-cut differences. And I'm just asking for clarification about, usually in these studies, blood's drawn at each vaccination time point, and so that information might have been available to the work group, but just wasn't presented to us.
So I think the clarification is that the 0 and 6 month, I believe that data has been shown and possibly at the last meeting, but there is a response, some response after the second dose. So if you're doing the 0, 1 to 2 month and then 6 month schedule, you will get some protection earlier than if you do the 0, 6 month schedule. And that's why the preference was for increased risk, people at increased risk or at immediate risk use that schedule.
Other questions? Thank you. Would someone like to make a motion? Yeah. One more question, Dr. Maldonado.
Sorry, I was just thinking, I don't know if you've talked about this at all, but this is, what would there, in a situation where you have a high-risk person who's already been fully vaccinated and they're being put in a high-risk situation for some reason, and it's past the point where the antibody titers drop off, is that something to be considered at all, or will that be for another day to discuss?
Right, so I think that's for another day, but I think the next topic that we'll discuss is boosters for people at increased risk, and when would someone need to be reboosted if they were at increased risk?
Thank you.
Yeah,
Dr. Zucker.
Yeah, a New York City Department of Health. I just want to point out from a programmatic point of view that for our registries, this will mean that we'll schedule the MenB series at 0 and 6. So as long as that's understood, because there is a challenge of translating the ACIP guidance, into what our registries are going to tell providers to do. And most often they're going to listen to our registry, and they may not actually be stepping back and asking questions about whether someone's at increased risk. So as long as that's the intention, then I think that's fine, and we can actually translate that into actual guidance for the pediatric community. Thank you.
Thank you. Okay, good.
Just briefly, and Dr. Zucker may be able to explain, it was a good question. Actually, Dr. Hunter mentioned whether immunization registries would key the forecasting based off of the brand of the initial vaccine given, so that it would be keyed for 1 or 2 months if it were the different interval if it were Bexaro given first versus Trumenba given first.
Yeah, so most registries can do that. It depends if we get a lot number. and whether or not they've actually reported it as a particular product, because it could actually come in as a MenB, sort of, NOS sort of product isn't specified for us.
And that creates its own complications because this is one vaccine where we're not supposed to mix the brands. We are supposed to give two doses or 3 of the same brand of vaccine.
Thank you. Is there a motion?
Will there be data presented at the next meeting about Bexsero and sort of 06 versus 01? Or
Grace, talk into the microphone, please.
Apologies. I'm just asking about whether or not an upcoming meeting we'll see data about the Bexsero vaccine, just in terms of 01 versus 06, just because from an implementation standpoint, I agree with Dr. Walter that we can do this in an incremental way, but it would be helpful to know that the long term, where we're headed, it just would be useful, because I don't think it'll be otherwise practical for us to implement this.
Thank you. Dr. Friedland?
Yes, Dr. Friedland from GSK. Dr. Lee, those data have been reviewed by the work group in the past and, of course, by the FDA. The majority, in fact, the vast majority of all the clinical trials with Bexsero were with 2 doses, and 2 doses were looked at on different schedules, 1 month after, 2 months after, or 6 months after the first dose. In those situations, the percentage of subjects who achieved the protective titer of 1 to 4 was high regardless of the schedule. In other words, a very high proportion of subjects achieved seroprotection on the 0-1 schedule, and there's no increase in percentage of subjects getting that seroprotective level when given 6 months later versus 1 month later. And this was the rationale for the indication that the second dose of Bexsero can be given as soon as 1 month after the first dose.
I just clarify if it's possible for us to consider this a little bit more broadly, which is to think about it as, if you're at high risk, can you just do a 0-1 schedule, 0-1 or 0-1-6? And if you're doing this sort of for permissive routine recommendation, that we just do 0-6. Because I just think that from a clinician standpoint, it'll be easier for them not to have sort of confusing recommendations.
Do you want to comment on that?
I think it's something the work group could certainly discuss.
Dr. Friedland?
Yes, if I just may, Dr. Friedland again from GSK. When we speak to healthcare providers about the use of Bexsero in clinical practice, we hear that the flexibility of giving that second dose as soon as 1 month after the first dose is something that's can be very important. For example, if there's a student who comes in to their practice during the summer months, the family can complete the full series, both doses, over the summer before they head off, let's say, to college. So having that flexibility is something that we're hearing is quite important and is valued by the healthcare provider.
Dr. Middleman.
Hi, Amy Middleman, Sam. I just want to make sure I understand this, because the policy, what's written here is, for persons at increased risk and for use during outbreaks, preference for a 3-dose schedule. Does that mean the working group has a preference to only recommend the 3-dose schedule, or that the working group is going to allow for either one, but a preference for the 3-dose schedule? I just want to make sure I'm understanding, because I would love to be able to still have the option for those healthy adolescents who want 3 doses to be able to get 3 doses and not be limited to 2 doses.
Right, so the intent is for people who are at increased risk or during outbreaks that they would get 3 doses, so they could have that early protection.
So that is the recommendation. So for healthy adolescents, is it that the recommendation is 2 doses, or the recommendation is a preference for 2 doses?
Maybe I should just clarify that the work group doesn't make recommendations. So the working group is expressing their preferences for ACIP to make recommendations.
Right, but is there preference for a preference, or is there preference for... Do they prefer to have a preference?
I guess what I would say is that the vaccine licensure allows for clinician flexibility. I think that what we've gotten as feedback, and many of you have told me this around these recommendations, is that it's already incredibly confusing to have the A versus B recommendation and the 2 different vaccines and all the other places where clinicians are forced or have to know something in order to make a simple recommendation. And so I think there is a value I place on you all trying, if possible, to make a recommendation that is as simple as possible in this area in a space that's already really complicated. So the ACIP can do whatever they want, but I would ask for a try at some kind of simplicity here.
Thank you. Dr. Stevens?
So the working group spent a lot of time on this question and trying to wrestle with this question in terms of whether we're preferring a specific 0.6 or an option. We kind of came to the conclusion that we would create more confusion with a mixed signal in terms of you can do either one. And I think that the data would support, at least in the view of the majority of the working group, the 0.6 schedule for healthy adolescents not at risk in a routine administration setting. So I think that was and it's been a lengthy discussion on the part of the working group with a number of straw votes, as Jessica can attest to.
Dr. Kemp?
So a follow-up, I mean, where the rubber meets the road is insurance companies will or will not cover 3 doses for healthy adolescents. I guess that's an implementation issue. Is this the ACIP saying either are okay, but we have a preference for one? Or is it saying we're making well, it doesn't sound like we're making a recommendation for one. Is that right? Because I mean,
I think that the AHIP representative is actually not here, but on the phone, and I invite him if he wants to comment. Remember, there are 2 places where this hits decisions about coverage, and one of them is a VFC vote, which ACIP has the power to make, but it is purposely separated from your recommendation because it's meant to be a second step. And then there is ACA. ACA is set up so that it is driven by what's on the immunization schedule, which is different, separate, and a discussion that we'll have tomorrow morning when you actually look at the schedules themselves. It doesn't actually have to be on the colored bars on the schedule. It can be in one of the footnotes. But the ACA language says that what is covered is what's on the immunization schedule. And if AHIP wants to comment.
So let me push this a little bit and suggest that we actually put a motion on the floor because I think that might clarify this a little bit. Would anyone care to make a motion that covers these recommendations?
So this is
Stevens.
Thank you. I think the motion would be for persons at increased risk, and it's stated here, for use at increased risk or for use during outbreaks, a preference for a 3-dose schedule at 0, 1 to 2, and 6 months, and in healthy adolescents, a preference for a 2-dose schedule at 0 and 6 months.
I just want to be clear because what you said before was that the work group strongly preferred to just have the recommendation for the 2-dose schedule, but the way that it's written there, it implies that it actually is a preference and that either schedule would be recommended.
I think the word preference is the way the working group would come out with it. Maybe I overstated it, but I think the word preference is what we agreed to. Jessica can comment on that, but I believe that's where we ended up.
That's right.
This is a uncharted territory for us a little bit. We've had this issue around preferences previously, and it is very difficult in the implementation phase when it's stated as a preference. And so I think we need a little guidance perhaps.
I actually think that maybe one of the problems, and we're coming up on our break, so maybe we can do it during the break, is we typically put up language. And so I think it's a little hard to have this conversation virtually, but a preference does imply that you all are okay with any of those groups getting either schedule. And you're right, we have been concerned about the language of preferences because it's very hard for us to implement. And so if where ACIP lands is you think either schedule is acceptable for either high risk or healthy adolescents, but express a preference, you certainly can say that. Again, it goes to this question of how is a clinician supposed to interpret that? And, if you really can't decide, then of course, go ahead. But this is back to my value of, it is incredibly already difficult to articulate what you all meant by a Category B recommendation. Adding another layer of complexity on top of also the 2 different vaccines is going to make this hard.
Dr. Lee?
May I put a different proposal on the table?
Yes, because it hasn't been seconded. So.
Okay. I would propose, I think what's throwing us off here is a specificity for the vaccine product, and I guess I'm not used to thinking of it that way, at least for me, under the MenB-FHbp up there. And so I would propose that the vote be about, for persons at increased risk and for use during outbreaks, a preference for a 3-dose schedule with one product or a 2-dose schedule with another product, but with the intent of it being in a shorter time frame. And you can work on the words. And then when given to healthy adolescents, a preference for a 2-dose schedule with either product. Does that make sense?
Dr. Kemp?
Alternatively, I think it should be a recommendation, because I think this is just problematic. And I think we're compounding the degree of complexity. I think that particular statement should be a recommendation.
I amend it to recommend instead of preference. Gotcha. Okay, now I gotcha. But I just want to flip it around in terms of the framing.
Dr. Belanger?
Yeah, not to beat a dead horse too much, but I think there's a difference between recommendation and preference, and I think it'd be most clear for clinicians if ACIP says that we recommend a 2-dose schedule for healthy adolescents.
May I suggest, again, if we're coming up on a break, if you finish this conversation, we can perhaps draft some language in the however long you can give us for break and then put something back up. And David, is that, yeah.
Dr. Moore?
I would just say that operationally, while I appreciate the scheduling idea of bringing things together, since these are 2 products that can't be interchanged, unlike all the other products that can, it does simplify things, where you can't mix a Trumenba and a Bexaro schedule. It does make sense. It isn't that much more complicated to make a specific FHbp recommendation in this case, because the other stands alone. But I recognize in terms of the schedule, it is nice to work on the timing, but here I think the recommendation could be more narrow.
Dr. Middleman, and then I think we will take the advice of our subject matter experts and take a break to craft wording for this. But Dr. Middleman?
Middleman Sam, I just want to, I appreciate the now not being a preference or a preference, but a recommendation. But I just want to make sure I understand that if it is up there for a healthy adolescent to have a 2-dose series, that it can still be on the immunization schedule, that a 3-dose series would be okay and therefore be covered if there were healthy adolescents who would like a 3-dose series.
Okay, I'm trying to see Melinda over there. So Melinda, this is correct, right? The schedule tomorrow language of the footnotes could still give flexibility. We can, Melinda is saying that, the discussion tomorrow around the schedule still gives you flexibility about what to put in the footnotes.
And may I also add that there's always language in all of our statements that if you get a dose at the wrong time or you did get the second dose at 1 to 2 months, that you should receive that 3rd dose to complete your schedule because you didn't get that dose at the right time. And so we have that sort of backup safety net for people to be fully protected if they didn't get that second dose after 6 months.
Yeah, and I appreciate that. It was just, there was a little bit of a question when Dr. Kroger mentioned that may actually penalize the provider in some cases in terms of coverage, but I don't know.
Okay, can I suggest that we take a half-hour break right now, be back at, that'll be 3:45, and at that time we'll have some specific wording for the motion and we can vote on the motion and the VFC. Thank you.